Hello YouTube, this is Sam from Tigers Schooling. In this session of the video, we're going to talk about the gluconeogenesis. Alright, so what is gluconeogenesis? It's actually reverse of glycolysis except few steps. Like, it's not exactly the reverse of glycolysis. Like, there are few steps that are not in the gluconeogenesis. But gluconeogenesis is something we call the making of glucose from non-carbohydrate molecule. What are those non-carbohydrate molecule? If I'm going to name them, they would be lactate, glycerol, amino acids, your pyruvate, and the list goes on. All right, so the major substrate, you can say, the major substrate which we put in to make to which go through the process of gluconeogenesis are lactate, glycerol, amino acids, and your pyruvate. So what is gluconeogenesis? We talked about it. It's the making of glucose from non-carbohydrate molecule. So what are those non-carbohydrate molecules? I'm again repeating them. It's lactate, glycerol, and your pyruvate molecule. Alright, so how does that happen, the gluconeogenesis? For that, I have to explain something else. Few things to keep in mind, that gluconeogenesis occurs in liver. Like 90% of your gluconeogenesis is actually occurring in the liver. If you're mentioning that one, it will be 90% in your liver. But for your 10%, it will be adrenal cortex and your kidney. So we'll mention only kidney. Alright. So this is the process. So this is the gluconeogenesis. Now let's move further. You know what happens in gluconeogenesis? In glycolysis, like comparison to glycolysis, we have to skip 3 steps. We have to create our own 3 steps. Like, let me explain it first. Remember this guy, glycolysis we talked about? If you haven't watched our lecture, please make sure to watch our lecture on glycolysis before, because if you don't know any idea or anything regarding glycolysis, you better watch that video, because you are not going to understand the gluconeogenesis. Both are related. So if you remember, in the glycolysis we made pyruvate molecule from the glucose. What happened? There were a lot of reversible reaction, but there were few reactions that were not reversible, so they were irreversible. So for example, converting the glucose from glucose 6-phosphate was irreversible. The second irreversible was this one, fructose 6-phosphate to fructose 1,6-bisphosphate, and the other one was phosphenyl, converting the phosphenyl pyruvate into the pyruvate molecule. You see that? Like 3 steps of glucose we have to skip. Like, look, in gluconeogenesis, what we have to do, we have to go from pyruvate to the glucose. But in glycolysis, we went from glucose to pyruvate. But in gluconeogenesis, it's opposite. We go to pyruvate to glucose. It's all similar steps, except 3 steps. We have to modify these steps. Like this one, from converting from the pyruvate, because we're going against it. So we have to convert the pyruvate into phosphoenol pyruvate. This guy is going to take another step, another idea, another path. So that's why this step has to change, this step has to change, and this step has to change. So 3 steps has to change of glycolysis in order to maintain the gluconeogenesis. So if we're going to talk about, so what we're going to do first, we're going to first convert the pyruvate into the oxaloacetate, then we're going to convert into the phosphoenol pyruvate, and this phosphoenol pyruvate normally goes through the process, like remember this one, if you have phosphoenol pyruvate, you know it can be irreversible, so we convert this one to that one, and to that one, until we reach this guy, fructose 1,6-bisphosphate, which is not reversible, so we have to use another enzyme. You see that? Look at here, 1 fructose 1-bisphosphate, fructose 1-bisphosphate. It's irreversible here. So we use another enzyme. That one, I'm just going to talk about the glucogenesis in just a bit. So this one, and we will convert into fructose 6-phosphate, and then again goes through a process and convert into glucose 6-phosphate. And again, this one over here, it's irreversible. So we need another pathway. Let me talk about it. So first of all, what we have to do, we have to make 4-step reaction. So we have to introduce 4 steps. Now, first step would be first step. Look, we're going to do it in the 4-step, normal 4-step, but it's the same 10 steps, but we have to add up or understand the 4 steps that are different from the glycolysis. So for number first step is the carboxylation of your pyruvate to oxaloacetate. Oxaloacetate. So what happens? Look at here. Look at here. It's very easy. You see that? Look, this is your pyruvate molecule. First, you have to convert your pyruvate molecule into oxaloacetate. Remember this one. Clear? Cut and clear. Glycolysis we knew occurred only in cytosol, but this guy, gluconeogenesis, occurs at two places. That is your mitochondria and your cytosol. But remember, only this step, by converting pyruvate into oxaloacetate, only that step occurs in your mitochondria. Everything else is in your cytosol. In the last step, we'll be talking about the part of cytosol in which it occurs. This step is going to occur into the ER, or you can endoplasmic reticulum. So converting glucose 6-phosphatase into blood glucose, this guy occurs in your liver and kidney, and this is going to occur in the endoplasmic reticulum. And this guy converting pyruvate into oxaloacetate occurs in your mitochondria. Everything else, your cytosol. Got it? Very easy. This one into your endoplasmic reticulum. This one, in converting pyruvate into oxaloacetate, only occurs in the mitochondria, and everything else in your glucose. So our first step would be carboxylation of pyruvate to oxaloacetate. In order to do that, what we have to do, we have to convert this guy to this guy. We need an enzyme called pyruvate carboxylase. So what happened in this reaction? So this carboxylation actually occurs. We use a substance called carboxybiotin. So this carboxybiotin give its carboxy part to this reaction, and biotin goes away. Like it separates and gives its carboxy part. So this incorporates into itself, and this pyruvate becomes the oxaloacetate. So this oxaloacetate, we know this guy is on our mitochondria. So we want to take it out of the mitochondria. How are we going to do that? We're not going to take it as it is out of the mitochondria. It is not going out of the mitochondria as it is. So what we have to do, we have to convert this guy into malate. Remember malate? It's a salt. Remember, the malate is a salt, or you can say a salt or ester of malic acid. So we can convert this oxaloacetate into malate, and malate is going outside, because this malate is going outside, and again that malate is going to look, first oxaloacetate convert into malate, and malate as it goes into the cytosol, it is again going to convert into the oxaloacetate. So malate is only a way to get out. Like when you want to go outside, for example, if you want to go a party, for example, you're going for a party, like if you're wearing a tux, all that thing that doesn't look like you're going for a party, they won't allow you, the guards won't allow you inside. But as you dress up, you look nice and clear and gentleman, they will allow you. So this guy is only changing its clothes. Its oxaloacetate is converting into malate and to enter into the cytosol, which is a party house, and again as it gets inside, it changes its clothes. So it's again converting into oxaloacetate. So from oxaloacetate to phosphoenol pyruvate, we use an enzyme called phosphoenol pyruvate carboxylase. But one thing we have to remember, that this reaction, converting the oxaloacetate into the phosphoenol pyruvate, occurs, where does it occur? It occurs in the cytosol. Remember, when we convert the phosphoenol pyruvate into the pyruvate, we use an enzyme pyruvate kinase, but it's different. Like this reaction was one step, but here we used two steps. Like we converted the pyruvate into oxaloacetate and oxaloacetate into the phosphoenol pyruvate in order to reach this guy, phosphoenol pyruvate. So this is it. The second step would be this one, by converting the oxaloacetate. Like if I'm going to write down the second step, I'll be saying conversion of your oxaloacetate into your phosphoenol pyruvate. That's it. Very simple. How does that gonna happen? We're gonna add up GTP. So if you're going to add up GTP, and again you lose the word GDP, and again you also lose what we call the carboxy group. All right, you lose the carbon dioxide in the form of carbon dioxide, and again you get the phosphoenol pyruvate. So number 3 reaction, as we go further from the phosphoenol pyruvate, remember from phosphoenol pyruvate till the fructose 1,6 bisphosphate, like till here all the reactions are irreversible, so they occur as it is. Like if you go backward, we only go backward. Remember this glycol, we are going backward, all the reactions are irreversible, same. So as we reach fructose 1,6 bisphosphate, this reaction was irreversible, so we're gonna do something else. What we're gonna do, that would be our third step, that would be dephosphorylation. That would be dephosphorylation of your fructose 1 bisphosphate. What does that mean? You are going to remove the phosphate group from your fructose 1 bisphosphate. So how you gonna do that? Very simple. You're gonna add up this fructose 1,6 bisphosphate. What you gonna do? You're gonna add up 1 water molecule into it. You add up 1 water molecule. As you add it up, this guy kind of lose its pi molecule and it's converted into the fructose 6 phosphate. You see, we added water molecule, its loss is by using an enzyme fructose 1,6 bisphosphatase. Remember this phosphatase, it's a phosphatase, it's a different. And the other by converting, last time we converted fructose 6 phosphate into fructose 1,6 bisphosphate, it was phosphofructose kinase. It's a different one. This here, it's a different one. We use fructose 1,6 bisphosphatase to convert the fructose 1,6 bisphosphate into the fructose 6 phosphate, and pi group is removed because last time it was on carbon number 1. So carbon number 1 phosphate is gone. We just left it. All right, now let's talk about the 4th step. That is a really funny step. Like we got till the fructose 6 phosphate. From fructose 6 phosphate, it's a normal reaction, it's an irreversible reaction to go from, because it's an isomerization. So from if you go from fructose 6 phosphate to glucose 6 phosphate, it's normal, it's irreversible, it's a isomerization. So till here we reach to glucose 6 phosphate, but here an interesting thing occurs. What happens? This glucose 6 phosphate has to convert into the blood glucose. So how does that happen? So remember, the 4th reaction would be the conversion of glucose 6 phosphate into the blood glucose. So conversion of glucose 6 phosphate into your blood glucose. What you have to do, you have to remember a few things. Your glucose 6 phosphatase is only present in your liver cells and to lesser extent in your kidney. So only these tissue can release free glucose into your blood. So it is very necessary that this conversion only occurs in your liver and kidney. Where does it occur? Like we have to remove the phosphate group in order to make it the glucose. So how does it occur? This reaction actually occurs in your lumen of endoplasmic reticulum. Remember this one just by heart. This reaction, conversion of glucose 6 phosphate into blood glucose, this guy is only going to occur in your lumen of the, what I said, lumen of your endoplasmic reticulum. And finally, as this occurs, we're finally getting our blood glucose. All right, this was very easy concept regarding the gluconeogenesis. Let me give it a quick review, just a quick review. What we have to do, we have to follow 4-step reaction, which is called gluconeogenesis, which is kind of opposite to the glycolysis, but not exactly opposite, because we have to bypass the irreversible reaction. How are we going to do it? We're going to first convert the pyruvate into the oxaloacetate, then oxaloacetate into phosphoenol pyruvate, and that phosphoenol pyruvate, as it goes on, it is converted into fructose 1,6 bisphosphate, and that one fructose 1,6 bisphosphate loses one phosphate molecule and converted into fructose 6 phosphate, and that fructose 6 phosphate is again going to normal process and converted into glucose 6 phosphate by that isomerization, and that glucose 6 phosphate, this guy, finally in your lumen of endoplasmic reticulum, going to convert into the blood glucose only in your liver and kidney. 90% your liver and 10% is kidney. That's it regarding the gluconeogenesis. And one thing more I haven't described here, you have to go through this step reaction. This is very easy. For normal 1 glucose molecule, we need, as a substrate, we need 2 pyruvate molecule. Remember that in that reaction, only 4 ATPs are used, 2 GTPs are used. Remember, 1 GTP, I have shown 1 GTP. You might be thinking, what the hell is that? But remember, we are going, this is a 3 carbon, 1 molecule. So we are using 2 pyruvate molecules. So that's why we have 2 pyruvate, 2 GTP, like 2 pyruvate, so 2 GTP, 4 ATP, we're using different places. and 2 NADH. So NADH was actually used over here in this reaction by converting the oxaloacetate into the phosphoenol pyruvate. So over here the NADH was used, and finally 6 water molecules. And this reaction finally leads us, these are all our substrate, all our inputs, all the substance that are required. This will convert into the pyruvate, 2 pyruvate, 1 glucose, 4 ATP, 4 ADP, 2 GTP, 2 DPP. Again, this 2 phosphate from here, 4 phosphate from here, we get 6 pi, and 2 NADH loses 2 hydrogen, so that's it, 2 hydrogen and 2 NADH plus. That's it regarding your gluconeogenesis. Please make sure to visit our website, that is www.tigerschooling.com, and make sure to be with us, and see you again. Thanks for watching.